Let me, besides Dr. Valentin Fuster, who's professor and physician-in-chief and legend in cardiology, is designated by the American College of Cardiology, our own chief of service, editor of Journal of American College of Cardiology and Hearst Cardiology. We have Dr. Narula, who is also world-renowned for his expertise, having been the founding editor of JACC Imaging, current associate editor of JACC Imaging, world-renowned for his pioneering work in atherothrombosis. We have three other really distinguished members of the Sinai institution. Carlos Cordon-Cardo obtained his MD from Autonomous University of Barcelona in 1980, PhD in cell biology and genetics from Cornell in 1985. He was vice chair of pathology and associate director for infrastructure at the Comprehensive Cancer Center at Columbia College of Physicians and Surgeons, and also created the Division of Molecular Pathology at Memorial Sloan Kettering. He currently is the Irene Hines Given and John Laporte Given Professor and Chairman for the Mount Sinai Health System Department of Pathology. He has been extraordinarily important in this fight against COVID because of his work with the assay, which Dr. Kramer and he have pioneered, which is both enzyme-linked immunoassay. ELISA is a common laboratory platform that can measure antibody titers, and it has been extraordinarily important. We are also honored that Adolfo Garcia-Sastra, PhD, a leader in the study of viruses, who was elected last year to the National Academy of Sciences, has honored us by his presence. He's director of the Global Health and Emerging Pathogens Institute and the Irene and Arthur Fishberg Professor of Microbiology and Medicine for Infectious Disease at the Icahn School of Medicine. His outstanding contributions to his field include developing reverse genetic techniques to reveal molecular basis of the influenza virus, which led to the recreation of the extinct pandemic influenza virus of 1918 and paved the way for improved influenza virus vaccine. So you can clearly see the link why he has been so important. He was one of the co-signees of a public statement for collaboration on COVID-19 vaccine development that was released by the World Health Organization. He has also been extraordinarily involved in coming up with a vaccine for fighting the pandemic, and not only this one, but potentially future ones which may evolve. Our other outstanding member of the Mount Sinai faculty who have joined us today, and we're privileged by her presence, is Dr. Gopi Patel, who so many of us know because of her leadership and hands-on performance during the pandemic. She's Associate Professor of Infectious Disease at Icahn School of Medicine, and has been instrumental in leading and designing protocols against the pandemic that we fight every day. We're privileged by their presence. And if I can, I see Dr. Satra. Is Dr. Cordon-Cardo on the call? Good morning. Dr. Patel? Okay, both were confirmed. If I do see that Dr. Adolfo Garcia-Sastra is on the phone, correct?
Hello, yes.
Thank you so much. Can you tell us a little bit about your work with the virus? And I want to quote something that you had said recently, that some viruses are very easy to make a vaccine for, and some are very complicated. It depends upon the specific characteristics of how the virus works. The next line in that article, not quoting you, but potentially flowing from that, it seems that COVID-19 is on the difficult end of the scale. Can you tell us a little bit, enlighten us about the vaccine formation. We know that there's 70 different vaccines in the work. We know that Oxford is starting one. There's 2 companies in China that are starting human work. They've tried it on monkeys and now trying it on human. Tell us a little bit, really enlighten us from your expertise about how do you go about identifying how to start the adequate virus vaccine and what the challenges are specifically with this one. Thank you.
Yeah, sure. So in terms of a vaccine against COVID-19, most of the approaches, and rightfully so, they are focused on getting a neutralizing antibody response with antibodies against the spike protein, which is the receptor binding protein of the virus. And that's what most of the vaccines that are working in humans are based on, the induction of neutralizing antibodies against the receptor binding protein of the viruses, whatever is a live attenuated vaccine or whatever is a recombinant vaccine. Many of the vaccines that are successful in humans, the main correlate of protection is neutralizing antibodies against the receptor binding protein. And that's the reason why these vaccines are all focused in the spike. There's some other vaccines that they take also into account potentially induction of cellular immune response against some other antigens, but I think right now the main focus is the spike and getting a neutralizing antibody response against it. Now, the antigen is clear. I think what it varies between the different vaccine approaches is the platform of vaccination. And in the past, there have come novel strategies for vaccination, and that's why we have so many different vaccines that are into consideration. Some of them are more traditional ones, like platforms that are based on inactivated viruses or into live attenuated viruses that have been used in the past for different viruses. Some others are more novel, like the one, for example, of Oxford, which is based in an adenovirus vector that has been used in the past, for example, for Ebola or for some other diseases, or the RNA vaccine that is been in clinical trials, also from Moderna. The main thing is, are these vaccines able to induce in humans a neutralizing antibody response against the virus? And is this neutralizing antibody response successful in containing the disease? And how long does it last, this particular response? And that's where there is still uncertainties, and it needs to be tested. With the caveat that testing might be quite difficult because of the conundrum about what you do with the phase 3 efficacy trial, in which in theory, in order to approve a vaccine, you need to vaccinate a large number of volunteers and follow them up whether they get infected or not infected as compared to a control. But obviously, there's some considerations about how this is going to be able to be done, because you would like also people not to get infected, obviously, or at least been treated properly. And you would like it to do it in a place where there is a lot of infections. If it's done in the clinical trial in a place where there is very little infections, because for whatever reason there is now lower amount of infections, then it takes a long time to figure out whether the vaccine is working. And I think it's there where the main problem lies. Now, in terms of my lab, we are working also in other experimental vaccines. None of them have been advanced yet into humans, but we hope that at least one of them. So I'm working in a live attenuated vaccine. I'm working in a vector also vaccine with a different vector from the Oxford people that we have been using in the past. And we are working also in a recombinant protein vaccine, which are based actually in the same antigen that it was mentioned before, that was made by Florian Kramer. So that's more or less what we are right now in terms of the vaccine arena.
Can I ask one question from Adolfo, if you don't mind, Dr. Goldman?
Sure, please go ahead, Dr. Narula.
So Adolfo, I hear specifically for the antibody test that you and Dr. Kramer have created, and also for the vaccine, you are going after that particular part of the S protein, which probably includes that linker, which is very specific to COVID-2 as compared to COVID-1 or bat coronaviruses or pangolins or whatever else it is. Is it really necessary to go after that, or is it possible to go after the other domains of the S protein, which are common with COVID-1 and others, so that you can come and kill the virus, even if that linker part is left? I mean, would that independent linker left in the human body, would that be still pathogenic?
So in terms of the as you say, is focused on the spike protein, but there is an area in the spike protein which is the receptor binding domain that is clearly a target of neutralizing antibodies because it will prevent binding to the receptor. That's something that we will be testing. So we have already getting approved for using hamsters, which seems to be a good model, at least not only of replication but of some disease with COVID-19, because mice they don't get disease and they don't get infected well due to the receptor, but hamsters do. And one of the first experiments that we are planning to do in hamsters is specifically this one. We are going to compare different versions of the spike, membrane bound, secreted spike, cleaved spike versus uncleaved spike, and receptor binding domain to see which one induce higher levels of neutralizing antibodies and are able to protect in the hamster in order to advance a specific antigen into future studies.
Thank you. We have a lot of questions. What I'd like to hold off on those. You know, how long do you think it's going to take? How do you confirm that some immunity is actually going to be protective? There are so many different questions, so please stay on. I previously introduced Carlos Cordon-Cardo as a professor and system chair in pathology, and he has now joined us. Thank you very much. Can you tell us a little bit about your developing the testing, what the challenges have been? There have been so many articles critical of false positives and false negatives. And it seems that the tests that you and Dr. Kramer have developed have been one of the most preeminent accurate type of tests, which are going to allow us to move forward. Thank you very much.
So when the pandemic started, we as an institution have been very fortunate to have a very strong academic department of microbiology, and Dr. Adolfo Garcia-Sastra is approved of it. We have a department of microbiology that has made important discoveries and developments through the years in the area of influenza. And the reagents were ready in here, thanks to the work that they have been doing for many years and what they were doing more recently, Adolfo being in China, Florian interchanging data with international colleagues. So probably because of all of that, the Department of Microbiology at the basic level was able to produce reagents that were very unique. And these reagents at some point, when we started discussing it, were going to be critical for patient management. And this is one of the things that this institution does well, translational research. We are real in translational research. We have terrific colleagues in basic science. We have doctors that I have not seen in many hospitals that I worked through my career. And the Department of Pathology, it's a very large department academically and clinically. We are probably the largest department of pathology in the country, doing over 100 million tests a year. So we had a good deal of platforms that were able to translate these basic assays into clinical, regulated, FDA-approved, New York State Department of Health approved tests. So we started with the detection of the viruses with a series of platforms. And from the very well beginning, we wanted to create something different. We were not just here to test. We were not just here to take pictures of H&E and have a collection of slides. We were here to translate data into knowledge and then to use this knowledge in order to better manage our patients. So we decided to do an end-to-end solution. The laboratorians call an end-to-end solution when you try to look at the disease from all of the angles. And in some areas, we are more experts than in others in the context of entire patient management. So we took the opportunity to say, let's do it well. Let's make sure that we test in the beginning and how we are going to test for diagnosis. So for diagnosis, we decided that we were going to do not only a vital detection, a qualitative detection based on a PCR assay, but that we were going to try to stratify the patients doing a viral load, which very few people have done it from the very well beginning of the disease. So not only we had a yes or no certification of the presence of the virus, but also a viral load that allow us now to stratify patients that are probably going to do poorly, and at the same time work with our researchers to make sure that they had the critical samples that were needed in order to see if there was going to be a new mutation or to be able to produce phylogenetic studies. Now that we have the disease diagnosis and the disease stratification in a good space, what else can we do? How we can follow these patients? So we went into a series of discussions trying to understand the two main areas of the disease, an area that is more immunological, that deals with antibody production, that deals with cytokine storms, that deals with a series of infiltrates and a series of developments in different organs, multi-organ disease that needed to be taken into account. So we put in place with Dr. Florian a test for the antibody that is very unique. Why it's very unique? Because with the bank that the Department of Microbiology has created and the bank of the Institute of Personalized Medicine, we were able to have true positives and true negatives. True negatives are those patients from which we had zero from 2016, 2018, pre-COVID era. So we were able to have tests that have had a good deal of robustness. The assay of Florian is also based on two steps. A qualitative step first with the binding domain, with the RPD, but then we come to a second step that is semi-quantitative. We are able to produce a titer. We are able to, like with the virus, we are able to put the patient in low, intermediate, or high titers. We did that with the blessing of the New York State Department of Health because we wanted to use individuals that they are antibody rich in their blood for potential plasmapheresis. So by the way, we are the group that has put more patients in this protocol probably in the country. That two steps, that qualitative and semi-quantitative, which we call in the laboratory orthogonal tests, tests that go in parallel, produce a great deal of specificity. As a laboratorian, I always care about two things. Is the test sensitive and is the test specific? And is the predictive value critical for patient management? And as much as I like sensitivity, in this case, sensitivity is not as important. Why? Because sensitivity deals with false negatives. To make a long story short, sensitivity is how accurate you are going to be while rendering a final result in avoiding false negatives. In this case, if I give you a false negative result and you are wise, you are not going to expose yourself to risk, hopefully. And even if you are, it's a false negative, which means that you are positive. So probably you're not as much at risk. And our test is very sensitive. Many tests are very sensitive in the market. Our test is over 94%. And the more you test, the better the test gets in the sensitivity. So we are not talking about sensitivity here. We're talking about specificity. Why specificity is critical in this assay? Because specificity deals not with false negative, but with false positives. So if I tell you that you are positive and you are really negative, and you feel that that you can now confront the disease because you have immunity, and you go into the front line, chances of being sick the first day are very high. And chances of then bringing that disease home or to the other colleagues at work are also very high. So we want to test in this specific situation to be as specific as possible. And maybe because of this double redundancy that we have and how we have orchestrated this test right now is 100% specific. We have done thousands of tests. I'll show you the data later if we have time. Many other tests don't have that accuracy. And even if a test is 95% specific, which in the laboratory world, it's a good test, in a million population, it means that 50,000 individuals will be falsely classified as false positives. 50,000 in a million. We are talking testing millions of people. We cannot afford a mistake. We may have not been the first to produce this assay, but we want to be the first to be right guided by science, being thorough, and knowing which are the important applications and when to do it. We also learn that if you test very early, the antibodies are not there. So we learn when is the best moment to test. And this is not going to be only important for the present time to open the gates of the society, but it's going to be also very important when vaccines come and to see if individuals have been obtaining that immunity that we hope we are going to have. Finally, the department also has been very involved as an academic department in performing autopsies of diseased COVID patients. Something that very few institutions did at the beginning, and we were one of the first to jump into it. We knew how important that was going to be. And with Adolfo and a series of colleagues, we have been able to produce a bank that I think is very unique. We have an autopsy room that it's quite modern in the context of airflows and negative pressure. So we have been able, with the right protective equipment, to produce complete autopsies. When you open the brain, you aerosolize, the potential viruses that are there. And we have unfortunately seen that is the case, that it's there. We have done over 60 autopsies. None of my colleagues, thanks God, have been, sick because of it, or we could attribute to that. So I think that we have learned how to do it in coordination with the microbiology and clinical colleagues. And we are accumulating data that reveals that this is a very different disease and that we have a lot to learn. But the opportunity to learn is here in that translational mode that we established from the beginning.
That was tremendous. There's so many questions, and I'm going to keep everyone on hold for a moment because I want to reintroduce Gopi Patel, who's on the line. We have the unique opportunity to have an expert who's developed the test, who's on the line. We have an expert who's developed and working on the vaccine on the line. And we have Dr. Patel, who from the beginning has orchestrated the infectious disease control response, which has protected us. And compared to other institutions, we've had a very low incident of healthcare workers being infected, I think a large part due to her efforts. And what I would like to pick your brain, Dr. Patel, again, thank you for joining us, is tell us a little bit about the responses you see, particularly if you can comment on the medication responses. We've seen so many conflicting reports about hydrochloroquine, azithromycin, steroids, and then obviously we're going to talk about something that other experts, Dr. Cordon-Cardo has been involved in plasma. But if you can address some of the medication responses, do you think that any of them have been affected? Which ones? And where do we go next with medical response? Dr. Patel.
Good morning. Thank you for having me. I think in terms of medications, we're still in an era of we don't know what we don't know. In a lot of efforts across the world, there's been an interest in translating what is seen in vitro in terms of anti-inflammatory or antiviral, and Dr. Garcia-Sastra can correct me if I'm wrong, because he knows the science much better than I do, in terms of lack of randomized clinical trials. And so, and with hydroxychloroquine, we always knew that it could prolong QT. We added azithromycin based on a small observational cohort study in France. And then we learned that we knew the combination would possibly cause cardiotoxicity or prolonged QT into torsades. And we don't know if that has contributed to some of the mortality in the ambulatory setting. Mount Sinai Hospital has always been careful about making sure we did not recommend that combination in the ambulatory setting. And now it looks like preprint data, not yet peer reviewed, from many, many institutions are suggesting.
Guys, we brought all these people.
Can we please have everyone on mute except our panelists? Thank you.
Are suggesting that we may have caused more harm. So increased mortality rate out of the VA in South Carolina, I believe it was North Carolina, some evidence of prolonged QT, although no mortality at our cousins at NYU. So I believe toward the end of this week, we'll be pulling back on recommending hydroxychloroquine. We're definitely not recommending it with azithromycin, but we're waiting for some New York State data to be released. In terms of remdesivir, there is some data that has leaked to the press suggesting that trial data confers some better outcomes, but it is unclear, and it's unclear when in disease, whether it's early disease or later in disease. And the thing is we didn't have people present when they were early in disease, so many individuals are presenting quite late in disease. In terms of the immunomodulators, because of the high levels of IL-6, there was a preprint or press release that came out of France about possibly tocilizumab and the sarilumab, which is an anti-IL-6 receptor immunomodulator. Some data is coming out about whether or not that has been helpful from the clinical trials. But I think we're in an evidence-free zone, and we're going to learn more as we compile data and clinical trials data is being released. Steroids, I think, remains controversial. So if you talk to our pulmonologists and intensivists, there's very little data for ARDS. There is a desire to use steroids in the setting of what may be the consolidative or organizing form of ARDS. Obviously, Dr. Fuster and many of you on this call have been involved in looking at anticoagulation, and is this even ARDS at all? So I think there's more to come. I can't tell you there's a silver bullet. I think we wait anxiously to hear about the convalescent plasma, upcoming hyperimmune globulin, and whether we'll get the clinical trials data for remdesivir and final data for sarilumab or tocilizumab.
Thank you very much. If we can just go back, because you touched on the plasma, Dr. Cordon-Cardo, can you tell us about your efforts and, again, why you thought that there would be benefit? How much did you decide to give? Is there a good basis to think that my antibodies would work in someone else? And do we know how much to give? So if you can give us a little bit of the background and some of the work that you've been a pioneer in, and then we'll ask Dr. Patel to tell us if she can give us a little bit insight to what she's seen clinically, whether there's been benefit so far. Dr. Cordon-Cardo, thank you.
Thank you. So it's clear that that some of us get cured when we get infected by viruses, and nature gives and takes. So I guess that we are exposed, but over the millions of years of evolution and the thousands of years that we have been here, we have been able to fight with our own armamentarium, and it's clear that that many of the treatments that we are using are not working at the level that we would like to, and we are understanding better the disease, and later I can tell you some of the mechanistic studies that we are doing, but the reality is that we do produce antibodies, and that some of these antibodies are terrific frontline soldiers. They are killers. How we know that? Because they are, again, assays in the basic laboratory that allow incubation of the virus, alive viruses, with antibodies, and you can see what we call neutralization. You can see that that our antibodies can really fight against these viruses, and that they can kill these viruses. So this hypothesis has been tested many times in the past with rudimentary plasma transference from one individual that was convalescent and did well to other people that were ill, and it worked. So it works also in other areas of infectious diseases, and it's good in areas in which viruses are not as critical and in the context of hypermutability. Some stability, it's good in here, because then the antibodies can be shared because we are fighting a common enemy. And this was our hypothesis. We realized that that all of the individuals that we were testing very early on were producing an immune response We waited in the context of convalescent individuals. We tested again, and we saw that some individuals that had low titers in the beginning were reaching a broad reach in antibodies kind of plasma that can be then translated into a strategy for treatment. We discussed that with the FDA. We discussed that with the New York Blood Center. We needed a colleague, a group that was able to help us in doing all of the volumes that we were going to need. We have a good chain. Again, it's like an end-to-end solution. You start by testing, you start by identifying, you call back, you realize that the titers are high. This individual wants really to help and to donate plasma for other clinically and critically ill individuals. And we had a great response. Actually, when we opened the door, the problem is that we had too many volunteers. A good sign of the society. Moments of difficulty, we help each other. So we sent some of the high titer individuals to the New York Blood Center, where they produce now the separation of the blood in the context of the needs, which are quite large, and producing that plasma that comes back to us in order to treat the patients that they are very sick. We join what was in the beginning a specific EIND, a clinical trial in which you enter a patient at a time, using protocols that were designed by Hopkins and Mayo, and we were very much in the beginning there because of the nature of the disease in this city, the number of young individuals that came and said we want to donate. We were able to put a lot of the patients in the clinical trial. And results are coming back. It's still maybe a little bit too early, but we have seen very good responses in some of the patients that were very, very sick. What else we are doing? Well, to take the plasma one by one, it's good. Historically, it's like the cell phones. It's the beginning of the cell phones. Now we have cell phones that they are very small and very smart, and you can do more things. So we are moving from an entire plasmapheresis to produce, separation of immunoglobulins, producing hyperimmune stocks that are going to address this disease even better. And finally, and hopefully, we are starting a study in which we are identifying young or under 35-year-old males. Females have sometimes anti-HLA antibodies, essentially they have been pregnant and they have had children. So we are trying to avoid other cross-contaminating antibodies. So we are taking young individuals. We are going to separate their B cells, which are their antibody-producing cells. We are going to clone these cells one by one. We are going to allow them to produce the immunoglobulin, the antibodies that they produce. We are going to test which one of these B cells, these clone B cells, produce the highest titer of antibody, and if this antibody again is neutralizing in a microbiology assay. And once we identify a B clone that has an immunoglobulin that is highly effective, we are going to genetically clone the DNA, the genetic code that codifies for that immunoglobulin, and we are going to try to mass produce it to have what is called a clone immunoglobulin that could be a terrific weapon into the future to help in specific stages of the disease at a different level that we are doing today with the entire convalescent plasma. So we have a plan, again, that goes from more conventional to more fully developed and scientifically driven products that we hope are going to be of help and that can save lives. Definitely, plasmapheresis in the past has saved lives, and in here we believe that it's saving lives.
That's fantastic. I want to ask a follow-up question of yourself, and it has to also touch on Garcia-Sastra's development of the vaccine. We've seen different immune responses, and people have spoken about age-related. What determines why one person is going to have a much stronger immune response than another? And also, is the antibody of each person almost identical, or is it individualized by that person's own immune system? So antibody from patient A may not be as strong as antibody from patient B, and is there any way of testing it? And that goes along with Dr. Garcia-Sastra as far as the immune response to a vaccine. Is there any way that it's predictable who's going to respond well? We know that multiple vaccines are used because age-related young, middle age, and older patients respond differently. So tell us a little bit about the immune response and how then you're looking at fantastic way of looking at the clone of certain B cells. But why do certain patients have a more robust response and some don't? Some who've had a very bad reaction have a low titer taken here at Sinai, and others have an extraordinarily high titer.
Well, if I can start at least with the clinical data, and I don't know if I can share the screen. Maybe I'll try. If yes.
On the bottom, yeah. If you touch share screen on the bottom, do you have that?
Yes.
Yeah. If you touch that green arrow.
Can you see?
Yeah, you're perfect.
Yes. Okay. Perfect. So this is a little presentation that I prepare for today, and probably what is important is this is what we discussed, how we are doing the different assays, but what is important here is this slide. This slide tells you as of yesterday how many individuals we have tested for antibody production. And as you can see, we are over 12,700 individuals that we have tested. And because of the nature of the test, which we were not testing in the beginning, just sick patients, but sick patients at different moments, and some healthcare providers also, and some of our healthcare workers, sorry. We got around 60% of positive individuals. But of these positive individuals, essentially 90% are donor eligible, which means that they have, intermediate to very high titers. And that's good, because we are learning that that in a way capable of going back, into opening the gates. It seems like maybe people take some time. Some individuals produce these antibodies a little bit later. Some antibodies are some other individuals are very efficient in producing the antibodies very early on. But the reality is that we are seeing that essentially everybody is producing antibodies and good titers of antibodies without essentially any exception. So it is true that there are some data that it's a little bit upsetting, because we are hearing that maybe this disease is not producing an immunity. Clinically, I don't see it. I see many patients, coming out of the disease well, thanks God. And we are seeing many individuals producing these titers. So again, it's maybe when it's the right time to do these antibody tests, and to be able to do it linearly, to have some continuation, because again, we have seen that several individuals that started with low titers went into intermediate and high titers, and at the end of the day, most of the individuals that have had the disease, when they are convalescent, they reach very high titers.
Dr. Garcia-Sastra, please.
Yeah, thanks, Carlos. You are completely right. And anyway, you are going to always expect in humans a spectrum, right? Not everybody is reacting exactly with the same potency. That's why it becomes also very important, the quantitative assays and the functional assays that Carlos was also talking about. Because if you combine the treatment with a good assay that tells you exactly what is the amount of neutralizing antibody that you are putting into people, what is the amount of antibody and neutralizing antibody that are put into people, then you can interpret much better what is the data that it comes out of the trials. This also allows to follow up people to see what is the percentage of infection in the people that is being followed up if infections continue to be prevalent later on. And this allows then to determine correlates of protection based on a titer that you get in sera, which helps enormously also in terms of a vaccine, because then you know what titers you want your vaccine to achieve according to what is known about natural protection from infection. So everything is related. The important thing, as Carlos says, having robust assays that tells you more than just we have antibody or we don't have antibody, tells you the quality of the antibody that you have, the functional quality, and quantities the amount of antibody that you have, so that then you can interpret the data both in terms of natural infection or reinfection of people that have antibodies, as well as those of that you use in the clinic, and then you see how much they treat. This also allows to do animal studies where you can compare what titer is protective in an animal with respect to a particular sera that comes from humans. And that allows also to better interpret, for example, what titer you may get in vaccines, and are you getting the same protective titers with a vaccine as you get with natural infection when you put it into an animal. So there is very interesting things that needs to be done still, but all the tools are there. The main thing is the speed, right? Because we know that vaccine development takes time, and the virus is very fast, obviously. So that's actually the main bottleneck at this moment in fighting against this virus with the vaccine.
So just following up on that, why do you need to look at an infection rate after a vaccine? Why not just measure the antibodies? And how do you determine what degree of antibody response is protective?
Yeah, so that's the hope, right? If we can come out before all the clinical studies are finalized, if we can come out with a correlate of protection, then efficacy trials may not be required in order to get an emergency vaccine. But for that, we need to establish a correlate of protection. And I think it might be possible to do it according to well-planned studies about what determines right now with the people that are natural infection, what constitutes a correlate of protection in terms of antibody titers in these people. And correlates of protection, we need to distinguish between two types of protection. One is protection from infection, and there is protection from disease. And there are two different things. In general, it's more easy to protect by antibody responses in sera, the lower respiratory tract, which is most likely correlated with severe disease, invasion by the virus of the lower respiratory tract, versus the upper respiratory tract that is a little bit more difficult to protect from antibodies in sera. They probably require also some mucosal antibodies. And this is also something that is very important to establish early on, and that is something that is very important for the clinical trials of vaccines later on, that I think the primary outcome should be not protection from infection, but the primary outcome should be protection from more severe disease, which makes also more difficult the clinical trial because not everybody goes into severe disease. And that's, again, where it becomes extremely important to have a correlate of protection, because then we may bypass some of the nuisance that you get in terms of number of people that you need to test for a clinical trial.
Thank you. Maybe I can ask Dr. Patel and maybe Dr. Niebart, who I see. There have been probably over 100 patients who've gotten plasma infusions. Can you tell us any early results that you think one way or the other can tell us whether they've been effective or not, Dr. Patel?
I think they're looking at the early results now as a system. It's been over, and Dr. Cordon-Cardo can correct me if I'm wrong, over 200 patients. If we include Mount Sinai South Nassau here in our sister site that received convalescent plasma. The data, it's really early because you sort of need to follow them out. And I can't remember when we started. I feel like it was the late part of March. I want to say March 26th may have been our first collection, and March 29th may have been our first infusion, but Dr. Cordon-Cardo probably knows those numbers better. So we need to be able to follow patients out far enough to truly say, was this successful? Did they go on ventilators? Did they not go on ventilators? And did they manage to be discharged, or what was their outcome? So I think they're in the early phase of looking at matched controls so that we can really look at this scientifically and not anecdotally. But Eric Niebart has really been on the front lines caring for patients. I'd be very interested to hear what he says.
Eric, thank you. You have to unmute yourself, Eric.
Yeah, yeah. You know, it's very hard to know in a lot of instances. I think one of the things that's going to be very important to do is to looking at cases as to when we administered therapies and looking at therapies administered, you know, before the ICU and on admission to the ICU. You know, I think we all have differences of opinions and whatnot, probably because of our perspective and which cases we've been seeing. I think Dr. Fuster presented some data to us yesterday that anticoagulation is beneficial. As far as the plasma therapy, you know, I think we need to see the data. Obviously, I've had stronger opinions on steroids and where to use them, you know, and I think, and you know, we may talk about it more next week, is, you know, we need a lot more emphasis on early in the disease.
Thank you, Eric. Can I ask, again, a lot of questions, but Dr. Cordon-Cardo spoke about 60 autopsies. Can you address some of the issues that we're fascinated by with the potential benefit of antithrombotic therapy? Can you address issues relevant to us as cardiologists about what you're seeing in the heart, the microvessel disease potentially and macrovessel disease? We're obviously concerned, you know, have you seen any patients who've had coronary interventions and what their stents look like afterwards? Can you tell us about your thoughts about the autopsies, the prothrombotic nature of this disease, microvessel involvement, and then particularly focusing on the heart and the pulmonary arterial tree?
Sure. Again, if I can share a couple of slides. These are some of the findings. I have been preparing a few slides to share with you and some other colleagues because we are getting lots of questions, and I asked my team to slow down in doing autopsies and speed up in looking at the results, because sometimes you get so involved in the work. And this is what we are seeing, which is very, very, very worrisome. You see the share screen right now? So this is the first slide with the respiratory system. And as you can see, thrombus formation, it's seen both in the gross, in the macroscopy during the autopsy. And of course, this is very much revealed also during the histology studies. In the lung, we have seen this thrombi formation, not only in big vessels, but also in intermediate and small vessels. There is plenty of evidence with transmission electron microscopy of the fibrin deposit in the capillaries. And the damage that we are seeing at the histology level in the parenchyma is not only produced by the viruses with these gigantic atypical pneumocytes that they are, you know, the typical histological images of cells that have been infected, but we have electron micrographs that allows us to see that really the pneumocytes do possess the viral particles, and that the lung is severely affected by a very acute and chronic inflammation that produce hyaline membranes and also a small vessel thrombosis. Of course, we have seen the presence of viral and viral-like particles in the nasopharynx, but probably for this group also very important the fact that in the cardiovascular system, we are seeing myocardial damage. We are seeing myocardial damage from the point of inflammation in between the cardiac fibers in a context of like a chronic myocarditis. Damage to the fibers themselves, the presence of virus and viral-like particles in between the myocardial fibers that they are being disrupted. We have seen signs of acute infarcts, and essentially both in the heart and in the epicardial, we see quite a bit of cases an important inflammatory component and microthrombi that affect even the intermediate and the small blood vessels. Finally, the other organ that I know that Dr. Valentin Fuster always talks about it is the central nervous system, where we have seen something very similar with focal perivascular hemorrhage, infarctions and necrotic areas, and microthrombi in multiple vessels of all calibers, from large to small. We believe, or some of us believe, that this is a disease that is also critically affecting endothelial cell. This is a disease, compared to some other diseases that we have seen in the past in this area, that we believe that endothelial cells may be a target. And we have been able in brain capillary to see viral particles, and viral particles that produce a cytoplasmic vacuoles that is the beginning of then later on infections. And we have seen intracytoplasmic vesicles in neural tissue. So this is a disease that, that to our understanding of these early studies, may be quite a bit relating multifactorial to endothelial cell damage. So, there are essentially two main pathological postulates of mechanistic insights into what may be happening right now with these patients. One is what is called the immunomodulation damage produced by an infection, by in this case, the SARS-CoV-2 virus, that produces a series of damage. And we believe that part of this damage is in the endothelial cells as well as in some other cells, producing inflammation and a cytokine storm, for which we have now antibodies that can block it and some others, but a very aggressive disease. The other disease that mechanistically that we are also studying, because we think that is going to be important, is the hypoxia damage that produces an apoptotic mode that is at the cell membrane level and guided by ceramide that produces essentially the blood vessels to lose their endothelial cells, and in these rough surfaces, of course, thrombosis and other developments are going to start happening. So these are some of the early findings. We have plenty more. With Adolfo and a series of colleagues, we have been taking in all of these autopsies material for conventional histology, for electron micrographs, studies, body fluids. We are doing vital loads in each of the organs. In the beginning, as Dr. Navy was saying, we had patients that were very, very, very sick for a while. I mean, some of the first autopsies were, hours after the autopsy, and with individuals that have been very, very, very ill. So some of the organs were very affected and difficult to study. We have been able to have a family liaison that is helping us in what we call rapid autopsies. So we are doing now autopsies hours after that. The tissues are better preserved, even though, again, these individuals have been very sick and dying for days. So not easy to do some of these studies, but the compilation of everything that we have, it's starting to show up a picture that may not be exclusive of these different mechanistic insights that people are postulating, and that we may be able to have a better understanding based on the cadre of scientists and clinicians that they are going to be able to study this disease at a different level, I hope, based on all of the materials that we have and that we are very, very anxious to start sharing.
I ask you, can you determine whether the cardiac endovascular and endothelial damage is a primary injury, or how much of it could be due to the hypoxia and the lung disease? Because I guess that's one of the seminal questions in this disease. Does the hypoxia then induce other organ damage, or is it a multi-organ disease to start, and then progresses fastest in the lung, and then the other organs slower, but also indeed a very strong impact?
So I think that we are going to see a combination of both. I am a little bit surprised, to be honest with you, in the lack of inflammation in some organs, and I don't mean lack, I mean the paucity. When you look at the nasopharynx or you look at the lung, you see really a lot of damage, and in most of the situations, inflammatory infiltrates that we are now starting to subset, and but in some other organs we don't see as much of that. So, but we see a lot of damage in the blood vessels. So we don't see a clear encephalitis, but we definitely see blood vessel damage in the brain. So we were discussing that actually yesterday with Adolfo, and Adolfo was saying, Carlos, I think that that one is not exclusive of the other, and maybe that's what we are going to learn, that in some individuals there is going to be an endothelial cell damage that is going to drive the difficult disease, and in some organs then you're going to have also a cytokine storm. And it's for that reason that we think that looking at surrogate markers in body fluids may be very important. For example, if we think that hypoxia is critical and that there is a endothelial cell damage, and we see it, they are sphingomyelinase and ceramide and other analytes that we can study in the blood and in body fluids. So I think that that getting these reagents will be critical.
Thank you very much. We just have a few minutes remaining. What I'd like to do is just ask each of our speakers give a closing comment about how we move forward, what's next on the horizon in their area, and then I'll open up for a few questions if we have time. We definitely have to have this panel back to get follow-up. I appreciate so much you taking the time. So Dr. Patel, can I start with you for some closing comments and perspective?
Sure. I think we're all trying to figure out what's the new normal or the better.
Can I ask everyone to be on mute unless they're speaking? Everyone please be on mute unless they're speaking. Thank you, Doctor. Thank you, Dr. Patel. I apologize.
It's okay. I think all of us are trying to figure out what our new normal or our better normal is. I just want to thank everyone who's been on the front line. This has been unprecedented. We've seen amazing agility, creativity, compassion, teamwork, and a level of humanity I think we haven't seen in a very long time. And Dr. Cordon-Cardo sort of referred to that. So I think for infection prevention, it really is how do we open? How do we go back to what is normal? And how do we also continue the kindness and humanity that we've seen in the past 8 to 9 weeks? It's hard to believe it's been about 8 weeks that we've been at this.
Thank you very much. Dr. Garcia-Sastra, closing comments and perspective for future.
Yeah, I think we have covered a lot of grounds. I just want to add something which is also a very important studies that can be additional to the ones that I have mentioned, and that's infection in laboratory settings of organoids as well as primary cells to figure out what could be the targets of viral infection in order to understand much better what could be driven by direct viral infection versus what could be driven by indirectly due to, sorry about that, indirectly due to some hypoxia damage or some cytokine storm that may come additionally to the damage. And in this respect, we are getting a lot of different organoids, and we are starting to get very interesting data. And I think that's going to also contribute more to understanding the disease.
Can I ask you the most pressing question? When is a vaccine going to be available, in your opinion, which is going to be effective and immunize the herd?
Yeah, I think it's going to take at least 1 year. I think it's going to be very difficult to move faster. It may be taking a few less months than 1 year, but 1 year I think is the most conservative for the time that we can put in.
Thank you.
Unfortunately.
Very much. And Dr. Cordon-Cardo, closing comments?
Well, you know, amplifying the message of Dr. Patel, we have seen a terrific, you know, spirit of collaboration, very dedicated colleagues, 24/7. The labs have been amazing, and you know, we have been here to make sure that we had all of the developments that we needed it. So I think that we have the diagnosis now more or less in place. Now what we want is to move on to start sharing this antibody test, that serology test that can open the society. So we hope that between now and June, we are going to be able to test probably in the couple of thousands of individuals per day. We are ramping up with colleagues in the commercial space. In the beginning of June, we may be able to produce enough testing capacity with colleagues that will be in the millions. If that is the case, and if the assay is as robust as we believe, we hope that between now and the fall, we are going to have plenty of the community tested, so we can go back at least to shake some hands and to embrace and to some of our family members. And hopefully we are prepared for the next development, because this is in part here to stay, so we need to be prepared with the diagnostic modalities that we have been putting together. And of course, now what is critical is to see what else can help us from the point of view of the treatments, which have to do with the mechanisms and being, smart enough to identify how we can tackle, the secrets that the virus used to make us so sick.
That's fantastic. Can I ask Dr. Fuster or Dr. Narula for any additional comments?
No, I think within the context of the integration mentioned, which is beautiful, I have a question to the three of you, actually. And this is a hypothesis with three variables. The first one is, what is going to happen in the winter coming up? And the hypothesis is, well, this virus will not go away. The second question there is that perhaps the situation will be much milder. And here are the two variables I want to ask. One is because we are better prepared to address the issue now after we learn of all the failures. But the second is it possible that the virus at that time, if it is still there, be less virulent, less aggressive? So I have a sense of the virus will be there, but I have a sense of optimism because we will be prepared to address it, and then maybe the virus will be different than what it is now by itself. And I'd like to know your opinion.
Okay, maybe I address that, Val. So I think we have the opportunity to be better prepared for what is going to happen next. It's likely, not a given, but it's likely that there will be a second wave, because I don't think that there is enough natural acquired immunity for preventing that the virus circulate again in a way that could be very robust, like has been infecting in the past, in the near past. So it's very likely that the second wave occurs. But I think if one has more robust, more available diagnostic tests, it may be able to detect infections more early on. We may be able to know what people are already some levels of protection, so therefore they are unlikely to be infected. And if we detect infections early on, it's going to be more easy to isolate the infected people early on, so that will prevent at least such massive infections that have happened now. So the evidence that we have from the virus that have been sequenced in New York, which with the help of Hart van Bakel here in Mount Sinai were able to do, indicates that the virus was already circulating in New York as early as January 15. And it took 2 months before it was detected. The first case was detected, basically, almost. So I don't think this was going to happen. We have now the opportunity to find infections very early on. And if we find infections very early on and they are coming up, then we can isolate the people, and we don't need to go over all the problems that we had before, even though still there will be a lot of susceptibles. Now, in terms of the virus becoming less virulent, I think, or more virulent, I think there is no time yet for the virus to evolve so much. I think the virulence is going to change not so much because of the intrinsic characteristic of the virus, but as we are acquiring a natural immunity, then obviously the virus is going to circulate less in people that have natural immunity. And at the end, with the time, this virus will become a virus that infects mainly people that they don't have natural immunity, which will be the people that are born after this pandemic. And that will be mainly then the very young people. And that, fortunately, severe disease in these young people is a very rare event.
Dr. Patel?
I think to piggyback over what Dr. Garcia-Sastra said. This was a failure to test, and I think being able to test widely, easily, and inexpensively will help us control. What we would like to see, we're still in mitigation phases. We're still in the crisis and contingency use of personal protective equipment. We have an amazing lab capacity, but the rapidity is something that we struggle with in certain scenarios. So really, how do we apply these tests, apply them early, and go back to what we consider containment, which is we identify an individual, we quarantine all those close contacts, we test asymptomatic individuals and really thoughtfully bring them back. And obviously be able to treat, if we have treatments available, those who cannot mount an immune response, which is the idea of the hyperimmune globulin, and us getting more knowledge collectively in controlled situations about which treatments we have actually work and when to offer those treatments.
Thank you very much. Closing comment, question, Dr. Narula?
Thank you, Dr. Goldman. I mean, this has been a fabulous discussion. Dr. Cordon-Cardo, Dr. Adolfo, and Gopi. It has been amazing. The only one thing that I would like to comment on is, and that is something which probably if I have 5 to 10 minutes, sometimes during the next few weeks, if I would be able to spend with Dr. Carlos Cardon, would be to talk to him about the myocarditis part, which I still believe that it is not really a myocarditis. And the reason being that if it is truly going into the myocardium, we should have started to see the myocyte inclusions, which we have not seen so far. Endothelium also does not get affected, because if there is not much of viremia, as we know, if there was endothelium getting involved, we would have a real catastrophe. We haven't seen that. I mean, nobody would have survived if the endothelium was, its ACE2 receptors were getting involved. In the lungs, yes, but not elsewhere. So routes are different. When we talk about its reaching the brain, essentially probably the route is the olfactory nerve rather than the blood-borne phenomenon that is happening. So myocarditis is and most of these people who have extensive levels of the troponin release in thousands, and these people come back. Some of them have the Takotsubo type of a syndrome. Some people develop just myocardial depression. Look at their right ventricle, they just balloon out. And but these people come back. So what exactly are we seeing? There is a very specific likelihood of a cytokine-induced problems, plus the possibility that we are looking at the microvascular thrombi, which in some people really come back. And so essentially, we really need to investigate it further. So myocardium is, as we always have known about myocardium, it is probably one of the smartest organs that we have in our body. It knows how to protect itself. So this is what is happening in this case also, and we need to elaborate on that. Thank you.
Thank you very much. You know, we had probably 5 of the smartest people in the field, in their respective field, in this panel discussion, and I was honored to participate. I apologize for dominating a lot of the question time, but there was just so much, and we had such an excellent panel. We will definitely have this panel back. I just wanted to express appreciation for all the contributions that each of you have made to fighting this pandemic. Each of these people have been on the news in the front pages of the papers, and we were privileged to have them contribute today. So thank you again very much. Continued great success. Unfortunately,
Martin, I have 2 comments. I'm sorry. Please. The first comment, this is fabulous. This is unbelievable, the meeting we had today. These are 3 guys who really are in the forefront. And I just want to emphasize this, is how lucky we are to have these people. I mean, this is unique. And the second comment is, there has been so much criticism of Spain being the worst country in the world at least I want to comment that two of the panelists today are from Spain. So just to give a little bit of a sense that Spain is not so bad.
Aren't you
Dr.
from
Fuster,
Spain
3,
also?
not 2.
Fuster?
Dr. Fuster, 3, not 2. And we also have the Sagrada Familia in the background.
Thank you. Thanks very much.
Thank you.
Again,
Fantastic.
Thank you all very much. We did make an attempt to record this. I'll know whether it was successful or not, and we'll have it available hopefully. So again, thank you all very much. Have a wonderful day and be safe.
Thank you.
And thank you again. Be well.
Thank you.
Thank you.